on the beers.
Got Dan Andrews over here. All right, so I'm going to just move this cord because I know I'm going to trip over it. So firstly, thanks to Wade, I don't know where he went in the audience, for inviting me tonight. It's so fantastic to be able to present in front of actual real people instead of a screen on Zoom. I was invited, yes, I think that deserves a clap. I was invited here tonight to talk about how to hack the immune response. So we know how devastating COVID-19 has been last year. So I thought I would just put up a little world map to show how devastating it actually has been. So we can see over here in the dark blue countries, these are the countries that have reported over 1 million cases of SARS-CoV-2 over the last year. I have a pretty crazy stat. So as of 2 days ago now, there's been 116 million confirmed cases of COVID-19, with 2.5 million of these being deaths. And that's crazy to even conceptualise. So if I can sort of put it in a little bit of perspective and just pick out one country, being the United States. So I know it's a little bit of an outlier, but in 2001, on the 11th of September, 2,900 people died in the terror attacks. Currently, 3,000 people a day die of COVID-19 in America. It's crazy to even think about. And even if we think about World War II, around 400,000 Americans died in World War II. And currently over 500,000 people have died in America from COVID-19. So it's crazy, crazy, crazy how devastating this virus has been. But SARS-CoV-2 is not the only virus that's ever caused a pandemic. So if we look back about 100 years ago to 1918, the Spanish flu was devastating. It actually killed over 33 million people, which is crazy to think about because that's about 13 times what COVID has killed. Crazy. So we know that viruses can infect us. We know that they can kill people, cause all of this devastation. But what actually is a virus? So some people like to talk in the news about how smart viruses are, how they can mutate to infect new people. But I'd just like to remind everyone that viruses are not actually living. They don't have brains. They can't think for themselves. They're pretty much just a ball of RNA, DNA, protein, and lipid. And that's pretty much all they consist of. And obviously we know they can infect living cells. But how do they actually infect us? So if we take SARS-CoV-2 as an example, it's a respiratory virus, so we have to breathe it in. The virus goes in through our nose or mouth and colonises in our lungs. So it infects our lung tissue. And then we look into our individual lung tissues, we see the virus going in there. So when viruses infect individual cells, they actually take over our cells and replicate themselves. And then they're able to burst out of these cells and infect surrounding cells and surrounding tissues. And when it does this, it causes a lot of cell death. And this is most likely where we get a lot of the symptoms from viral infections from, so like pneumonia, for example. So when we're infected, we have obviously a peak of infectiveness. And then during this peak of infectiveness is where we have symptoms. But it's important to note that before we get symptoms, there's also a little period where we don't have symptoms, where we're at our most infectious. And that's usually how viruses spread so easily. It's because people don't actually know they're infected to start with. So I thought I would just put up a quick slide to show you 5 of the most, well, not most common viruses, but pretty common viruses. And I thought I would get a show of hands of who thinks that at least one of these viruses has a cure. No one? Oh, some hands in the crowd. None of these have cures. So some of them have quite good vaccines, so you have to be vaccinated before you get infected. Some of them have okay antivirals, but if we were to talk about HIV, for example, you have to be on those antivirals for life. As soon as you stop taking the drug, the virus is able to replicate at a massive level and cause symptoms again. All right. So we know how bad viruses are. We know how they infect us now. But how do we combat them? How do we fight them? So there's 2 main ways we can do this. So the first is vaccines, which I'm sure you've heard a lot about in the news recently. and the second one is antivirals. What's the difference between these two? And the simple answer to this is that they target different stages of our immune response. So when we're infected with a pathogen, it can be a virus, it can be bacteria, we actually have two immune responses that it sets off. We have a very early innate immune response, and then we have a later adaptive immune response. So our early immune response actually kicks off a few minutes after we get infected, whereas the adaptive immune response takes a few days to sort of ramp up. When we talk about being vaccinated, this is targeting the adaptive immune response. So there's been a lot of controversy in the news about whether vaccines work, what happens when you're vaccinated, am I going to be microchipped when I'm vaccinated? I'm here to say that vaccines definitely do work. Here's top 7 diseases that vaccines actually have nearly eradicated. So if we take one example, which is smallpox, about 5 years after the vaccine was developed and rolled out, this disease was eradicated. So no one now has smallpox disease. So I think that that's pretty cool to be able to eradicate a disease. If we're looking at the number of COVID-19 cases from the very start to 2 days ago, we see this gradual trend upwards until we see the vaccine roll out, and then we get a decline in numbers. And that's because, if we're coming back to this stat, 250 million people so far have been vaccinated against COVID-19. So I think that that is pretty cool to see such a drastic effect with just a couple of vaccines that have been made in such a short amount of time. So sorry about this very rough drawing. I was running out of time when I had to send Wade my slides yesterday. And I thought it was the best way I could describe it by scribbling it down. So, thanks. So how do vaccines actually work? So you would have heard in the news there's different types of vaccines, they target different things. You might have heard mRNA float around, protein float around, but I'm here to tell you the nitty-gritty. But before I do that, I have to tell you how our body makes a protein. So we have DNA, and DNA is the genetic sort of barcode for all of life. We have DNA in every single one of our cells except for red blood cells. So these cells are able to produce proteins in any of our cells. Before our DNA can turn into protein, it must be made into a more readable version, which is called mRNA. So this is sort of like a recipe for a protein that it gives your cell. This mRNA can then be translated into a protein, and proteins make up all of the structural and functional components of our body. So when you're infected with a virus or a bacteria or any kind of pathogen, they have proteins on their surface. And our body has a really, really, really cool way of knowing when a protein enters its cell that it's not itself. It sort of freaks out and says, like, what is this foreign object in me? That sounded really bad, but I didn't mean it like that. We'll take that back. So when we're thinking of the Moderna and the Pfizer vaccines, they actually use an mRNA vaccine. So they're using sort of the recipe to make the protein. They're not injecting DNA into you, they're not injecting part of the virus into you, but they're simply just giving your body the instructions to make this protein. So when we are vaccinated, this mRNA is surrounded by a lipid bilayer in a vaccine. And it's really important that we use a lipid bilayer because our cells are made up of lipids, and that way it can incorporate into your cells very easily. This mRNA is sent into your cells and it goes to the machinery that makes the proteins. Now, it's important to note that this mRNA does not go to our DNA. It doesn't get mixed in with the DNA. I don't know how many people have read comments on Facebook about how they're changing our DNA or anything like that, but it doesn't actually go anywhere near our DNA. It's then made into a protein, and because our cell can notice that this protein is not from itself, it puts it on the surface and it has a cry for help, like, help, this protein is not from me, this is foreign, I need help. And this is when the immune cells can come in, they can remember it, and they can produce antibodies. And antibodies are what are fast acting, and when you are exposed to the virus the next time, they're able to be expressed and that's what allows you not to be infected the next time. So that's how a vaccine works. But if we're looking at the other side of the immune response, which is what I work on, the innate immune response, this happens so quickly following infection. And this is what we need to target antiviral drugs to. So vaccines are fantastic for if you haven't already been infected. But what if you get the flu or you get COVID and you're in the first day or two of infection? We need drugs to be able to target that and to be able to not progress that into a worse disease. So, another fun diagram. So when we're infected with a virus, our virus will release its RNA or DNA, whatever kind of virus it is, and its proteins into our cells. These components of the virus are then picked up by our receptors, much the same way as the protein is picked up by immune cells during a vaccine. And then our DNA is able to go on and make some antiviral proteins. These antiviral proteins are very complex. They go on and do a lot of different things. They can fight the virus directly, or they can go up and keep running through this pathway to upregulate different types of immune proteins. This way, immune cells can come in and be in the area of infection and really fight this infection quite quickly. But if a virus, like COVID-19, it can hide from our immune response, and this is when we get severe disease. So what we're looking at is we target our receptors and try and enhance this response so we can fight this viral infection. So we target the receptors with synthetic drugs. And the synthetic drugs that we try and use are small parts of the RNA or DNA from the virus that might be detected by our own cells, or sometimes proteins. So it's important to remember that there's actually a pretty small window when it comes to antiviral treatments. So it's right at the start of infection before it gets too crazy and out of control. So we hope that when you take antiviral treatments at the start of infection, this viral load will then drop off and you won't get that serious disease and hopefully no deaths. So I'll talk like one slide about what we actually do, because it gets quite complex and I know it's sort of hard to understand fully. But when we're infected with a virus, what we look at is there's small little compartments within your cells. So if you think of a cell like the human body, we have lots of organs that do different functions. So within a cell, they have things called organelles or mini organs. They do different functions within the cell. And the organ that I look at is called the lipid droplet, which by its name, it sort of suggests it's literally a droplet filled with lipid. We have shown that some of these antiviral proteins actually live on the lipid droplet, and they need to live on the lipid droplet to keep running through this immune pathway and upregulate enough antiviral proteins to fight off the infection. So what we try and do is we try and manipulate this response. We try and add more lipid droplets into the response, or we try and target these proteins to enhance this response. And just so you know that I'm not lying, this is a picture I took on the microscope. They do look like a blob of of green. So I think my picture is pretty accurate. And we can tell that these proteins sit on these lipid droplets because of this yellow staining here. So the protein is in red and the lipid droplet is in green, and when they're touching, it becomes yellow. Simple, I don't know what you call colours. So why do we need this? So obviously I've talked a bit about how vaccines are so important, but I think that antivirals are equally important. So if you're already infected with something, you need a treatment that is going to be able to bring down that viral load so you get a lesser disease. I think it's really important also if we can develop novel antiviral therapeutics, because these might be effective across several viral families, whereas vaccines, as we know, they take ages to develop and they're very specific for a certain type of virus. So even when a virus mutates, it might still not be effective against it. But antiviral treatments, because they target your own immune response and boost that, they might be effective across several different viral families. So I think that's pretty cool. And I think that this is my last slide. So I don't know whether I went really fast, but this is our team that we work with. We work at La Trobe. There's some of them up the back there. So thanks for listening. Sorry,
Thanks,
I feel like I
Ebony.
went really quickly.
Very efficient. We'll take a few questions. One over there. What's the difference between virus, disease, and condition?
All right, so the virus is the thing I talked about that is filled with RNA, DNA, and proteins. The virus, when it infects your body, will cause disease. So the disease that you get is usually from your body fighting it off. So it will produce these antiviral proteins and things and cause like inflammation and heat, and it's called like a cytokine storm. So you actually usually get like pneumonia and things like that. So that's the disease. And what was the other one? A condition. So it depends what you mean about a condition. So like diabetes. So diabetes is more of like your immune, like an overreactive immune response, and several other diseases are like that too. So it's actually pretty similar to when you get infected, how your immune response sort of sets up this antiviral response. It's like a few different diseases have this where your body recognizes something foreign, although it might be your own body, and it produces this inflammation in your body. And so you get this disease from a condition or a disease thing. Does that make sense? Oh. I think I just rambled.
All very interlinked. Another question? We'll take one up the back and then we'll come to the front. Yeah.
You talked about the importance of getting in quick with antivirals before it sort of starts itself. How does COVID fit in the perspective of you don't have symptoms until you've got X amount, and by then you sort of got a race to not be too late? Is it harder or easier than other viruses?
How does antiviral fit in with COVID? How hard is it?
It's a really good question. So it is all about timing, and you don't want to leave it too late because the antivirals won't really do anything. I think when you start to get symptoms, it's not too late. It's about when the symptoms get really bad, it's probably too late. If you're talking in terms of like days, we talk about being infectious for about 14 days with COVID. I think if you were to take an antiviral treatment in the first 10 days, it's probably actually effective. And it also depends how severe your disease is. So if the disease is really bad, sometimes antiviral treatments won't actually do anything to help that if it's already gotten to that like stage, because it's already boosting your immune response, and that will cause further disease by giving you inflammation and things like that. So you sort of want to target it quite early, but it is quite hard to catch it early.
Early enough, but not too late. Okay. Up the front.
Okay, I'm going to preface this by saying I'm really just trying to learn. So if
No,
this comes
go.
up wrong, I don't mean it to be. So obviously with the quickness with which we were able to come up with a COVID vaccine, it leaves a lot of questions about why haven't we created vaccines for, you know, the other things that you've shown on your screen, HIV, etc. And there's obviously the mindset that because it's far more lucrative to sell the medicines to keep people healthy than it is to create a vaccine and eradicate it. So I'm just kind of curious on your take on that theory and you know, why you think we don't have a vaccine for some of these other things when there are so many things.
Okay, so we've been really quick with vaccines for COVID. What about the others? What's taking so long?
I think the simple answer to that is money. Government, please give us money. I saw a really cool thing on Twitter, like in the middle of last year, and it said people are concerned at how quickly we're developing a COVID vaccine, but they would be surprised to know that we could develop a lot more with a lot more funding. And it's so true. I think that is the main driver of why we can't develop things quick enough. And obviously there's been a lot of money thrown at COVID because it's costing them a lot of money to have people in hospitals. And I think that's the simple answer. I think there's a lot of viruses that are obviously a lot more complex than COVID. COVID is a relatively simple virus to study, and now that so many people around the world are studying it, putting their results online, it's quite easy to come up with things. But yeah, if I was to say it really simply, it is really funding and manpower behind it. And I think it's pretty sad that that's the answer, but that is the answer. No worries.
All comes down to funding.
It does.
All right. Over there?
How does one strain of coronavirus change its level more compared to mutated strain?
How does coronavirus mutate?
That is a very loaded question. If I go back to third-year microbiology lessons in university, so when a virus infects you, it replicates so quickly, and it makes thousands and thousands and thousands of copies of itself. Not every copy is perfect. Some of them will never infect another cell again. Some of them infect a lot better. And it's really like the survival of the fittest. And that's how we get a new strain, is like a tiny, tiny mutation because it's a mistake, and then it takes off into a new strain. That's like the simplest way to explain it, but it's the truth.
Okay. Strong mistakes.
Strong
Okay,
mistakes.
we'll take another question.
One of the earlier slides showing a bunch of different viruses and how it went from big number of cases to a small number of cases right now. And I think it was Rubella that was sort of down to 3 cases. Does that mean we finally got that licked? Or is it that's just projection and it's not just 3 people?
So I think that was 3 people that were infected that year. So it's an interesting question because there probably would be more people, right? You would assume that there's probably more than 3 people being infected, but I suppose there's not a massive amount of infection that we don't pick up on it. It's quite an interesting one, and I know it's a question that's been asked during COVID, especially in underdeveloped countries, and they're underreporting of viral infections. It's quite interesting. I feel like that's like a PhD all in of itself.
Another Nite talk. One more question. Oh, there's lots of people at the back. I couldn't see you. I'm so sorry.
It is really hard to see people at the back.
We'll take a question over there. Oh, I didn't quite hear that, but you did. Do you want
The
to?
question was, pretty much, do vaccines cause autism? If I was to go back, I think about 10 years ago, there was one scientist that published a paper about how vaccines cause autism. He made up the results, and that paper has been retracted. It's completely made up, the fact that vaccines cause autism, and it's pretty much being perpetuated on Facebook, and that's the only reason that exists as a concept. As I said, most of the vaccines that we use these days and all of the COVID vaccines don't actually go to our DNA, so there's no way that it can change our DNA or cause a disease like that. I think it's really important to remember that the vaccines that they're putting out, especially in 2021, are going to be completely safe. They've tested for more things than what they could think of.
Okay. We had another question from that table, I think. Yep. Yep. You go. Yep.
Now that your research topic is in the media all the time, what are your biggest pet peeves and how it's covered?
What are your biggest pet peeves about how your research being covered now that it's got so much media on it?
Oh, that is a very good question.
Maybe top 2 pet peeves.
The top 2. So I gave a talk at my old high school the other day, and someone came up to me and told me that they were an anti-vaxxer, even though I didn't ask. And they said to me, so you know that the virus is man-made? And I think that is probably my number 1 pet peeve, that there was like this conspiracy that the virus was man-made. I think that was definitely number 1. My number 2 would be not so much a pet peeve, but it's just maybe like the lack of education of people, or the lack of money put into science education and science communication, as particularly in, yes? Particularly in developed countries, when we have the money, we have the resources, we have the scientists for it, but we still don't think of it as important. I think that would be my second pet peeve.
Bring your friends to Nerd Night and learn more.
Absolutely.
Okay, we'll take one more question out the back. Are there any more at the back?
I think you got them all.
All right.
Wade's doing these ones.
Wade's going, wrap it up. Thank you very much, and we will be back in 10 minutes with speaker number 3. Thank you so much, Ebony.